Welcome to Advanced Data Analysis 2 with me, Erik Erhardt, Professor of Statistics at the University of New Mexico. In this video, we'll discuss Chapter 15 on multivariate analysis of variance. So this is multivariate ANOVA, or Manova, and the goal of Manova is to answer the question, are groups of observations, such as male and female or different species of plants, are they different in their multivariate response? In particular, we're going to look at an example here about turtle shells, the length, width, and height of these turtle shells, and ask, in this three-dimensional vector, are the centers of our observations different from each other for males and females? So this is an example that we looked at in a previous chapter, but we only looked at the females. Here we're looking at both males and females. So I'll read the data set in. Here is the tables for the data set. We have sex F, which is the baseline group, and then sex M, which is the male group. And as you look down these columns of length for females and length for males, you can see that I've sorted the data in this. I think I've probably sorted it by the length column, and that generally the females are larger in all three columns, length, width, and height. Let's take a look at the plot for the data, and I'm creating a scatter plot using the GG pairs from gGalley, and I'm also going to use a Scatterplot 3D to look at a three-dimensional scatter plot of the data. And on the left, let me zoom in just a little bit, and a little bit less. The first row and column is sex. Female is red, male is blue. And the key observation here is in the top left corner we have 24 observations for each group. So these bars are at the same height at 24. And that for each, the length, width, and height, you look at the spread and the center of the females versus the males. The females are more spread and their centers are closer to the upper bound for the males. So it looks like there are big differences between these two sexes for turtles. Furthermore, in the scatterplot matrices, you can see the very strong correlation between all three size measurements, indicating that the shells grow somewhat uniformly in all three dimensions at the same time. Though I'm noticing there's an interesting feature in this particular cell of the scatterplot right here, the length by width plot, which to my eye suggests that it's not quite linear in their growth, that as they get longer in the horizontal axis, they start by getting longer, more long than they are wide, or growing wide at the same time. And then at some point, right about here, you can see it starts to steepen, so that they're still growing long, but their width is increasing faster. So there's a relationship between their length and width that isn't strictly linear, and that as they get longer, the speed that they get wider seems to increase. On the right is a three-dimensional scatter plot, and let's look at a open graphics library version of this that we can rotate around. So we'll use the RGL library, the plot 3D function, and R seems to have changed slightly, and I'll show you a minor change in this piece of code here to identify the color. So we're going to plot for the x-axis will be the length, y-axis will be the width, and z will be the height, and then I'm specifying a color as sex. Now sex here, let me go to R and I'll drag the window over where we can see it. In fact, let me do an even better job. So the variable sex, I've already looked at it here. The structure of shells, we have sex is a factor variable with two levels. And what I'm expecting, so sex is a factor variable, so we know that it has numeric values but character labels, or I should say numeric levels, these 1s and 2s. So if we do as numeric for shell's sex, you can see that we've got 1s and 2s, and if we ask for it as a character vector, it will give the labels. So by default, historically in R anyway, when you did, if we go back to the notes for a second, if you do color equals sex, it would have picked up the numeric labels. But I tried that just a minute ago to rerun my code, and when I did that, so there's the line that we see in the, it says invalid color name F. So apparently now it's using sex as a character vector. So which means that I need to change my notes and add to it by saying as numeric around sex. Once you do that, it will create this plot here. So we have length on the horizontal axis, width on the axis going toward the back, and then height on the vertical axis. And it allows us to rotate it. Now we don't have a legend here for which group is which, but we know that females are larger, so they're the black points, and the males are smaller, they're the red points. It allows us to rotate the plot around. In fact, we can look, let's start at the origin. The origin is in this bottom corner, and look up the axis. Okay. So it's pretty clear to me that there's a good perspective right there of how different these two groups are. The center for the females is somewhere where my cursor is now, and then the red males are down here. So that's quite a difference. So it appears visually that there's differences between males and females. Now we're going to see how the multivariate ANOVA addresses this question. And in fact, Manova gives us, considers the answers to these two questions. First, are the population mean length, width, and height all considered all together the same for males and females? So it's a two-sample test, but with a vector response. And if they are not equal, then the interesting next question is, what's the combination of features? So what linear combination of these, of length, width, and height, is most responsible for the differences? That is, in which direction in this three-dimensional space is the difference between males and females most pronounced? And when we have the case of just two groups, you can imagine plotting the mean. Let's see. You can imagine plotting the mean for the females and then the mean for the males, and then connecting those 2 points with a line, and that's the direction through three-dimensional space that those two groups are the most different. And in this case, it looks almost like it's right down the this axis of correlation, though males seem to be a little bit below the females, at least in this perspective. So it looks like they are not quite as tall as females, too, and they differ up and down along the height axis. Okay. I encourage you to play with that three-dimensional scatter plot if it helps you better visualize these two clouds of male and female turtle points. All right. To describe Manova, suppose we have P features. In our turtle example, we have 3 features of length, width, and height. On independent random samples from K strata, in our case we have 2 strata, we have males and females, groups or populations, and let mu I prime, so the prime here is doing a transpose so that this looks like a row vector. I have some notational issues here, perhaps. Be this vector of features. So we have so I here is the ith population. So when I equals 1, that's females, and when I equals 2, that's males. Then we have our P features. We have 1, 2, up to P, and in our case we have 3 features. And so mu1, this mean is going to be the mean for the length, width, and height for group 1, which is females. And then mu2 will be for males, the mean length, width, and height for males. Then we ask the question, we ask the null hypothesis here is whether all the means are equal to each other. So it's the same null hypothesis, except rather than each mean being a scalar or a single number, it's now a vector. And in our turtle example, we have a 3-dimensional vector here for females and a 3-dimensional vector here for males, and that's it. And the alternative is the same, that all the means are somehow different, but we don't know how. All right. Assume that the sample sizes for the different groups are n1 and n2 up to nk. Those are our different sample sizes, so our grand total sample size is just adding them all up. Then we can calculate, then we've got individual observations, xij. So I here is the sample, so for example females, and then j indicates which individual from that sample. So there were 24 females, so j here will go from 1 up to 24, and xij1 is the length measurement. So we have length, width, and height for the turtle shells, those 3 measurements, and I indexes that we've got males or females, that's 1 and 2, and j indicates which individual within each sample that we have. And so these points really, this is just notation to describe this table right here. So here's the shells data set. Here are the 24 observations for females, and here is the length, width, and height measurements. So these first 3 values are sex is I equals 1, that's for females, and then j equals 1, it's the first observation, and then we have x1, x2, and x3, those 3 measurements. So that's I equals 1 for females, j equals 1 for the first observation, and then 1, 2, and p is 3, those 3 measurements. So just a way of notating, we need 3 subscripts because we have 3 different dimensions that we're looking at: individuals, samples, and which feature. Finally, we can calculate the mean, the sample mean for each group. So I is females and males, and this is just the mean over all the lengths, all the widths, and all the heights of the cells. And we also have a grand mean here over all individuals in the data set. We can calculate the covariance matrix over all features for the entire population by calculating the covariance matrix for each group and then taking a weighted average of those 2 groups. And finally, just as in analysis of variance, we have a Manova table. And it looks the same as analysis of variance table. We have between variability, that is, how different is each group, so xi, versus the grand mean, x. So that's the between group variability. It asks effectively how different are the means between the males and the females. And then there's the within variability. That's looking at all the observations. In fact, it's sort of summarized already by the covariance matrix. How different are the individuals within a sample? So think of the males. We have the plot for the males right here, the red points, and how much spread is there among the red points? In when we have one dimension, you can look at the histogram and see how spread that histogram is. In this multivariate space, you're asking effectively how spread is this cloud of points. And you can see right now that the spread for the red male points is less than the spread for the larger female black points. All right, and we also have an error, a source of error, which isn't represented in this table right now, but then the total variability. So the difference between what's explained by within variability and between and within variability, whatever's left over from the total is the error. All right, so I'm going to scroll down a little bit. Our assumptions are that we have independent samples. That is, each turtle was sampled independently, and the probability of one of those turtles being sampled didn't depend on which other turtles were sampled, and that they follow multivariate normal populations. That means that ideally our cloud of points would be like an ellipse, so that if you were to project the points in any direction, you'd get a univariate normal distribution. And similarly, we have identical variance covariance matrices. So this is like in a two-sample t-test from last semester, assuming that the variance for each of our groups is equal. Remember, when it wasn't equal, we'd use the Satterthwaite approximation for the degrees of freedom, but when they were equal, we could use this pooled variance. So here we're going to assume that the variances are the same for males and females. All right. All right, I'm going to describe this qualitatively. So the null hypothesis of equal population mean vectors should be rejected when the difference among mean vectors, as measured as the between mean squared matrix, is large relative to the variability within groups, as measured by the error mean squared matrix. So that is that if you imagine we've got two clouds of points, here they are. Don't imagine too much, here they are right here. So we've got two clouds of points, and right now we see that these two clouds of points don't overlap very much. And another way to summarize that is that there's within variability, which is effectively the variability within a cloud of points. So how much does the red cloud vary and how much does the black cloud vary? And then we have between, so that is the within sums of squares, is how we summarize that. Then we have between sums of squares. That is, if we calculate the mean for the red group and the mean for the black group, how different are those two means? And in this case, it looks like the means between the groups are quite large relative to the variability within the groups. And I'm concluding that because the two clouds of points don't overlap very much. You can imagine a situation where they overlapped a lot, which would indicate that the difference between the means would be small relative to how much variability there is within each cluster of points. So it looks here like we've got a big between group variability relative to the smaller within group variability. And it's that ratio that's going to give us an F-test. In fact, we're going to have 4 different types of tests in a moment that gives us a large F-test to indicate that there's a difference between groups. All right, I've got some numerical summaries, lots of numeric summaries, and all right, let's assess whether the assumptions are met. In particular, let's look at the standard deviations of each feature for females up top and males below. Now, my eye says that the females are almost twice as variable as the males. Do you see that too? we have basically 20. Up down here we have 10, 13 and 7, and 8, and almost 4. So it seems that the females are roughly twice as variable as the males. That's a little bit of an issue for us because we have the assumption that the variances are equal. Even maybe a little bit more information here. Here's the correlation matrix. So yeah, so I'm looking at, all right, so I've said that the assumption is that the covariance matrices are the same between groups. The two things that go into a covariance matrix are the correlation matrix, so how related are each feature, what are the linear relationships between the features, and the variances or standard deviations. So if you combine the correlations and the standard deviations, those two together gives you the covariance. The reason I'm not looking strictly at the covariances is because I think the standard deviations and the correlation matrices are easier to interpret than covariance matrices. So already we've seen that the variance seems to be twice as big for the females. The second part of that is the correlations should be the same. So the first matrix is the correlation matrix between all the observations, including males and females all together. And why did I do that? Now I don't know why I did that. I had an idea going into it, and all right, the main thing to focus on is we've got females and males. Let's see how these two correlations matrices differ. And it looks like the females have slightly stronger correlation between their features than the males. Part of that is likely due to the larger spread of the observations, but so it looks like effectively females are more variable and they have stronger associations between their features than the males. So this is a little bit of an issue for us. Things that we could do to remedy this is to consider transforming the features to try to get their variances to be about the same. If I look at the Scatterplot matrix or 3D Scatterplot, if you were to take the log transform, for example, for length, width, and height all together, it would shrink all these points closer to the origin, which is in the bottom left-hand corner, and that's going to improve the spread, the difference in spread of the points, I believe. So that would be a thing to try. All right. The major problem with having differences in variances is that you basically inflate the variance term, and it makes it you have a little less power to detect differences between groups. Here we're not going to, that's going to be the primary effect of having differences in variance. So it probably isn't going to work against us in terms of doing this hypothesis test. So let's continue forward, having recognized that there's a little bit of a difference in the covariances between the two groups. All right, I'm going to plot the data in terms of their principal components. This is going to give us three axes that are uncorrelated, and I'll do a scatterplot matrix of those principal components. And it looks like no specific principal component really separates these three groups. You can see how much overlap there is. And it looks like there's more difference, however, in the first component than in the other two. All right. For comparisons with the Manova that I'm about to do, let's look at the three analysis of variances, one for each of the features. So if we consider just length, so here's sort of a fancy way to do a bunch of univariate ANOVAs. Here's my linear model. I'm going to make comparisons by sex, and I'm combining these three columns, and when you do that within a linear model, it actually fits three separate linear models, one for each response term. So here we have length by sex, and we find that there's a big difference between males and females, about -22. So the t-statistic is -4.5, which is pretty large for length, as a p-value that's 10 to the -5. For width, as a t-statistic of about 4.7, same order of magnitude for the p-value. And for height, t-statistics of -6 and p-value 10 to the -8th. So it seems here that actually height is the larger difference between these three. But we can do even better when we use Manova. Let's start by checking our assumptions using some. So we already looked at it visually, and we did compared some covariance matrices and so on. But let's use the multivariate normality test package to perform the Shapiro test for normality. And if we ask whether the females have a normal distribution, p-value of 0.01. So this suggests that they are not quite normal. For males, p-value of 0.13, not enough evidence to suggest that they're not normal. And I wrote this little graphical assessment tool for multivariate analysis, where we calculate, well, I'm just going to, maybe I'll just say that I compare a Mahalanobis distance to a chi-square distribution quantiles, and when you put in the data set for females and males, it produces a plot like a QQ plot, which says that the Mahalanobis distance should scale with the chi-square quantiles and follow this line. And you can see this downward curvature for females, whereas the males follow the line better. Okay, so it indicates that there's some deviance from normality for females and not enough evidence for that for males. In our plot here, when we can find the right orientation, there was a little bit of curvature in the female points, and I believe it's that curvature that is the difference from normality. Maybe this isn't the greatest projection, but these points sort of curve up a little bit, the black points, and I think it's that deviance, that difference from normality that is causing all this, or that difference from linearity that's causing the difference from normality. All right, finally, multivariate test statistics. So we've got 4 of them, and I'm going to favor one over these 4 for performing the multivariate ANOVA. There's the Wilks lambda, which is the most commonly used statistic for testing differences. It considers differences over all the characteristic roots. Characteristic roots, those are our eigenvectors and eigenvalues. Not an important thing to understand right now. And the smaller the value of the Wilks lambda, the larger the differences between groups. Pillai's trace is another one. It also considers all characteristic roots, and it's a little bit more robust than Wilks lambda from the difference from model assumptions. So if your model assumptions are validated, Pillai's trace may be a little bit a better choice. Hotelling's trace is a third, and then Roy's greatest characteristic root is the one that I'm going to focus on because it relates to the next chapter, chapter 6 on discriminant analysis, and most interestingly, it will actually tell you the direction of the biggest difference between groups. However, when your assumptions are violated, it's highly sensitive to that and can give you fairly inaccurate results when your assumptions aren't met. But when they are, it's the most powerful test among these 4. Finally, here is the Manova, and I'm going to show you running this using two different ways. This lowercase Manova is built into the base R. You column bind the responses on the left, tilde sex is the categorical variable on the right, and then you can ask for a summary of the, here man.sh is, man is for Manova, and sh is for the shells dataset, and I'm asking for the Wilks test. And so here's the output for that. Here's the Wilks statistic. This is the p-value for the test. And you can see that this p-value is larger than any of the individual p-values we saw above. The smallest p-value we saw above was times 10 to the negative 8. And the other two were 10 to the negative 6, I think. So when you can combine all three dimensions, you can find even greater differences. If we use the conditional autoregressive CAR package and do capital M Manova and look at the summary, it gives some output here. We've got a sums of squares for the error. So this is the sums of squares matrix for the sums of squares error matrix. On the diagonal are the sums of squares of error for each individual feature, and we have a matrix for all of them combined. We also have a model sums of squares matrix, which is called hypothesis, and it is functions of these error and hypotheses matrices that give us these test statistics for these four methods. And if you want a summary of these four, and if you've got Ron Christensen's book, Advanced Linear Modeling, it's his green book, he shows the derivation of each of these. And you can see when we have just two groups that, in fact, the p-value is the same for all the tests. All right. Furthermore, Roy's test locates the linear combination of features that produces the most significant one-way ANOVA test of no differences among groups. That is, it finds the linear combination, so the direction that if you were to do a one-sample a one-dimensional ANOVA test along that dimension, that's the direction that gives you the strongest evidence for differences between the groups. The only difference is that because you have used the data to suggest, here it is, use the linear combination being suggested by the data for which direction, Roy's root also does a correction for basically letting the data tell you which direction is most different between groups. All right. So if I pull out the hypothesis sums of squares and the error sums of squares, those two matrices that we saw above, and I perform E inverse times H, that matrix multiplication there, and ask about the eigenvectors and eigenvalues of that matrix, it gives us the eigenvalues and the eigenvectors. All right, let me scroll up for a moment, because I believe this eigenvalue, 1.584, is the test statistic, 1.584, for the Roy's root test. In this case, it's also the same for the Hotelling-Lawley test. Not a big deal. I'm scrolling back down, and these vectors tell us the direction that is in the direction of the greatest difference between the two means. In our case, where we have two means, you can think of having a mean for the females, a mean for the males in this three-dimensional space, and connecting those two dots with a straight line. It's that direction that is the greatest difference between the two groups. All right, so direction 1, D1 here, I've just plugged in. I've just plugged in this first column, 0.215, 1014, negative 0.97. That's the linear combination that gives us the biggest difference. And the other two don't really count for much. And we'll see that in a moment. All right, let me plot the data now in terms of these three new variables, D1, D2, and D3. Those are our three directions indicated by the characteristic vectors. And I'll plot that using G-Galley. All right, and this is perhaps the coolest result. D1 is the direction that the two groups differ in the most. And you can see, either maybe in the box plots, how little the two groups overlap. Same thing in the histograms and in these density estimates. So if the data are multivariate normal, and then D1 is the direction that indicates basically all the difference. D2 and D3 should indicate basically no difference between the groups. And you can see how much they overlap, basically completely. So we've taken the three-dimensional shells data, and we found one direction that addresses the differences between the two groups, and the other two directions, there's no difference at all. We scroll down, we're almost to the end here. If we look at the test for just the D1 linear combination of features, how much it differs by sex, you can see how strong that difference is, times 10 to the negative 11, which is much stronger evidence than we saw in the using length, width, or height separately. All right, and that is the end. There was one more idea here about, I don't remember where it was. Here. In these eigenvalues, 1.58 is sort of how different the males and females are in this D1 direction, in this linear combination. These other two characteristic roots, or eigenvalues, indicate how different the two groups are in the other directions. And you can see that these are both 10 to the negative 15. They're basically zero. So all the difference is captured in that first direction. That, I think, is super sweet in terms of multivariate analysis, where you could have high dimensional data, you could have 15, 20 dimensions, and this test is going to give you the direction that the groups differ in the most. In the next chapter, we'll follow this up with discriminant analysis, which is all about finding directions that that groups differ by, and then designing rules that allow you to discriminate between separate populations. It basically gives you a decision rule for classification, which is the following chapter. So all these ideas are connecting. And that is the end. I apologize this was a longer video, but you got it all in one.